Oh, hello there. I didn't see you. Well, I guess none of us are seeing a lot of each other these days because of the COVID crisis, but it's so good that everybody is staying at home and flattening the curve. And well, that brings us to 3MT, the reason we're here today. Trent University's 3-minute thesis normally would be done very publicly in a large space, but this year we have to do the home edition. So welcome to 3MT this year, and we have 8 very intrepid grad students who are willing to share their research with us. So this is going to be a very exciting evening. I'm Dr. Michael Eamon, the principal of Catherine Parr Trail College at Trent University, and I'm so honored to be with you this evening to host 3MT. Now, this event couldn't happen without so many people working together. It is a signature event for Trail College, but it also receives the support of all the colleges at Trent University. It also receives the support of the Office of the Provost, of External Relations, and of course, of Graduate Studies. We have a very special partner this year in the Trent Graduate Student Association, and we'll hear more about them a little later on in the program. Most importantly though, the Academic Skills Center at Trent University has been a driving force behind this 3MT event, and particularly Dana Capel, who has been with us since the very beginning and helps bring this event together every year. Now, this event started actually in Australia. Now, we have many ideas of Australia, of kangaroos, of koalas, of a lot of beer drinking and other things like that, but these are all stereotypes. Australia also has some wonderful universities, such as the University of Queensland, which brought us 3-minute thesis to begin with. And 3-minute thesis is really interesting because what it does is it tells grad students to take what is years of work and condense it down into a 3-minute presentation. How do you take your life's work so far and bring it down into 3 minutes? It's no easy challenge, and that is what we'll be doing today, looking at our 8 contestants and seeing how they bring their work to life. But before we begin, we should cover the rules. As you know, 3-minute thesis is an international competition, and all across the world, students have to follow the same guidelines, and they are only one single slide can be used in the presentation. No additional electronic media, no animations, no music, no elements of any kind can be added to those slides. As fun as it might be, no props, no top hats, no canes, no laboratory equipment can be used in the 3-minute presentation. And again, the presentation has to be 3 minutes exactly. It has to be also the spoken word with no poems, no rapping or songs as used. And most importantly, no notes. Well, now that we've talked about the rules, we should probably talk about how we're going to judge this year's 3MT competition. In the past, we've had a celebrity panel of local judges who have helped us determine who are the best 3MT presenters. Unfortunately, this year, we don't have that celebrity panel of judges, so we're going to call upon you to be our all-star celebrities. We want to hear from you and hear what are the viewers' choices for 3MT. So how are you going to do this? Well, go to our website, www.trentu.ca/3MT. On that website, you will see this video, which you can share with friends, family, and colleagues. Also, there'll be a big button that says Vote Now. Click on that button and vote. You'll be taken to a special online form, but be careful, you only have one chance to vote. Also, you have to vote by May 24th, that is Sunday, May 24th, by midnight. That way we can tabulate all the different online votes and then do a presentation later on the 25th, that is Monday the 25th at 7 p.m. to find out the winners. We have 2 Viewers' Choice Awards. We have the Provost Viewers' Choice Award, and we also have the TGSA, that is the Trent Graduate Student Association Viewers' Choice Runner-Up. Great awards, great recognition for our presenters. Please look at the clips. Please be honest and fair. I know you may want to vote for friends and family, but give them all a chance and take a look at them and vote. Again, www.trentu.ca/3MT. Now, we might put up a little slide right here, and that will give us the criteria. There are 3 main criteria that our professional judges would use, and we would like you to use those criteria as well. One is communication style. The second is comprehension, and the third is engagement. So please judge fairly upon comprehension, engagement, and communication style, and most importantly, have a good time. Are you sitting comfortably? Then we should begin. 3MT, if there's a silver lining in this COVID crisis, 3MT can be a little bit more relaxed. We can sit down in our homes, pour our favorite beverage, have some snackos, eat some popcorn, and really enjoy the presentations that are about to happen. And for me, it is an honor to introduce these wonderful students. As principal of Trail College, I get to see many of them in their daily lives, and I can count them as friends and colleagues, and it is really exciting to see your friends and colleagues shine. So let's see some great student presentations shine right now. Without further ado, we'll start with Roya Gahramani. Roya is from Caledon, Ontario, and she's studying psychology. And we asked all the students to give us some bio tidbits that we could talk about so you get a better feel of the presenter. And Roya said that she loves horseback riding, she loves piano, and she loves to annoy her friends, which, if they're watching now, forget about the annoying bits and maybe just open your mind to the glory which is going to be unbridled potential, the use of equine-assisted learning as a resiliency intervention. Roya.
When I was a little girl, I spent a lot of time with a pony named Romeo. Now, Romeo had been through a lot of abuse and trauma in his past life before he came to us. And because of this, my favorite thing to ask him was, "Romeo, Romeo, why for art thou so stubborn?" I mean, seriously, it took about 30 minutes of blood, sweat, and tears just to get him to walk into the barn. Well, one day I came home and I went to visit Romeo. I had had a really awful day. My parents were fighting again. I was fighting with my best friend. I had failed an algebra test. And I looked at Romeo, I took 3 deep breaths, and I said, "I get it. Life can be rough sometimes." And you know what he did right after that? He walked right into the barn, no questions asked. Now that I'm in grad school, I have to ask myself, is there a way to emulate this feeling of confidence, this feeling of connectedness with another being, and this feeling of emotional regulation, maybe in other girls who have been through adverse experiences in their lives? Now, recently in the horse world, there's an up-and-coming intervention called equine-assisted learning. What this is working on the ground with horses, doing things such as leading them, learning to groom them, to learn about things such as boundaries, communication, and teamwork. This works because horses are prey and herd animals, so they're masters of mirroring our emotions and our actions back to us. This way we learn transferable life skills, and this translates to that sense of mastery, that sense of relatedness, and that emotional regulation that I felt that day with Romeo. And what do these 3 things make up? They make up resilience, our ability to tackle adversity head on and come out of it a better, stronger person. So we gave 12 groups of girls who had been through interpersonal trauma in their past the opportunity to participate in an equine-assisted learning program that lasted for 8 weeks. They went to the barn once a week and they spent time with these horses doing these activities. In order to measure their resilience outcomes, we administered surveys before the intervention, after the intervention, as well at the 1-month, 6-month, and 12-month follow-up periods. And now what we are seeing by investigating these surveys is that indeed their sense of mastery, their sense of relatedness, and their emotional regulation are increasing. But what we also did is collect feedback to see the real nitty-gritty of what is going on inside participants' heads. And this is maybe the most telling of all of our outcomes, of all of our results. It is so encouraging to see the girls write things such as, "I felt powerful, I felt respected, I felt like I was going to be okay." Thank you.
Wow, that was great, Roya. Thank you for leading us, starting us off with the first presentation. It's going to be a hard act to follow for sure. Well, next we have Jared Asser. Jared is a history graduate student, and he is a very interesting fellow from Oshawa, Ontario. Jared is interested in his thesis and looking at the Ku Klux Klan and their musical traditions. He said he started this by being interested in Soviet-era musical traditions and moving to the Ku Klux Klan. It's not necessarily the most direct line that I can think of, but I think it's pretty interesting how our lives twist and turn and how topics form. Jared says when he's not doing history that he likes to do yoga and go for hikes. So I feel that these are two hobbies that are very COVID friendly. So without further ado, let's hear from Jared Asser and his presentation, Hearing the Invisible Empire, Understanding Music and the Formation of an American Hate Group.
Hello everyone. Today I would like you to say hello to the famous Muncie Klan Band. In 1923, they crisscrossed the state of Indiana, performing in at least a dozen Klan parades. They were one of 40 such groups who staged hundreds of performances of Klan music between 1922 and 1925 in the state of Indiana alone. When I began my research, I wanted to understand where Klan music came from, as well as its influence on the early 1920s. What I discovered was a deep connection between the evangelical revival and the Klan's music. Klan musicians appropriated the form of the revival by adapting hymns and imitating its performance context. Now, at this time, churches were the most important facet of community life, yet they were divided by denomination, going bankrupt, and losing their place in an increasingly modern America. It is within this historical context that the Klan was able to present itself as a viable alternative religious community. As a result of their parading in Indiana, the Klan enticed 250,000 white Protestant men to join their organization, with estimates of the women's organization at roughly the same level. This translated into a voting bloc which engaged with politics outside of the norms of a two-party system. Parades quickly became political and expressed anxiety over immigration and education. Music mobilized Klansmen in support of the Immigration Act of 1924, America's first comprehensive law prohibiting immigration. At the local level, they mandated Protestant religious education in all public schools. Even more notorious was their slate of electoral victories. The Klan openly supported Ed Jackson in Indiana's 1924 race for governor. Klansmen wrote election songs and organized events in areas near where Jackson's opponents had recently been campaigning. As a result, they increased voter turnout by 20% and effected a mass crossing of the party line in favor of the Republican candidate Jackson. This victory was buttressed by the election of mayors and other local officials in local contests throughout the state. Beyond the KKK, my research tells of the deeply influential role of evangelical identity in American life and history, which gives context to the recent rise in rightist and conservative groups. Recall the singing en masse of Amazing Grace at a 2018 Trump rally. Furthermore, in both the 2016 and upcoming 2020 presidential elections, 1/3 of America's swing states have a devout evangelical populace. Thank you.
Wow, what another great presentation. Thank you so much for sharing that with us, Jared. And now we're going to get on to our third presenter, who is Josephine Esposto. She is in our ENOS, or Environmental and Life Sciences program, and she's from Hamilton, Ontario. She was telling me that she has always been fascinated by neuroscience, and she'll be talking a little bit about her research today. Outside of the world of neuroscience, she enjoys ice hockey and golfing and watching Leafs games, all things that perhaps have been put on hold. She told me that last summer she lived in Manhattan, so I'm pretty sure that she's happy that she's now back at home in that we're in the COVID crisis. But hopefully, here's to going back to Manhattan, here's to more Leafs games, well, maybe not. Here's to Manhattan, here's to golf and ice hockey in general, and here's to Josephine Esposto and immunotherapies targeting protein pathogenesis of ALS-associated protein TDP43.
Everything in this world is controlled by our brains. Our brains control our thoughts, our feelings, our pain, our hunger. Everything we interact with, including our own voluntary movements, are controlled by the brain. Well, what if we can control it? What if we could control our own brains? In a perfect world, we would be able to control everything from how our thoughts are processed to the ability to predict mistakes and fix them before they happen. In a perfect world, neurological diseases would cease to exist. In this world, we're getting pretty close to understanding just how the brain works, including how it makes mistakes. These mistakes are also known as neurodegenerative diseases. One particular neurodisease is ALS. ALS gradually paralyzes the body because the brain is no longer able to communicate with the muscles of the body that we are typically able to move at will. Our brain is connected to our muscles through millions of specialized nerve cells called motor neurons. These serve as our body's internal wiring and enable us to move as we choose. The brain sends a signal along these motor neurons telling a muscle when to contract. This signal is created by electrical impulses from the action of chemicals in our neurons. In ALS, however, motor neurons gradually break down and die. Researchers still don't know exactly what causes these motor neurons to die. However, gene changes or mutations in DNA have been successfully linked to ALS onset. My research focuses on one such gene change, one that makes a protein called TDP43. TDP43 is a naturally occurring protein in the body that regulates certain genetic processes. When mutated, however, has been shown to clump together and initiate toxic behaviors, leading to the onset of ALS. This protein may be toxic to other neurons and may initiate a cascade of damaged cells among brain tissue if mutated. In my research, I am studying just how TDP43 mutates from its original form to its toxic form. Once this aspect is established, I can then take this mutated TDP43 one step further, with the addition of larger, more specific proteins. These new proteins will specifically bind to my protein of interest and either enhance or prevent the mutations from occurring. If it prevents these mutations, this large protein could be the stepping stone to a therapeutic approach to the treatment of ALS. Either way, the addition of these larger proteins will illustrate key functions of the mutated TDP43 protein. This could be a novel approach to the future therapeutic treatment and cure for ALS. Thank you.
That was great, Josephine. You know, as a professor at Trent University, it never ceases to amaze me what students can do, the level of detail in their research, how articulate they can be in communicating that research. It is always such a joy to hear students such as Josephine. So thank you so much for your presentation once again. And talking about another impressive person, the next speaker that we have is someone I see at Trail College quite frequently, Juanita. Juanita Spears is in the Canadian Studies and Indigenous Studies program, and she really is an impressive person. She, when I asked her, I said, where are you from? She goes, well, wherever my hat is. My father was in the Air Force, and so I moved around Canada and, had many homes, but now she calls Bowmanville, more or less, her place of residence. Now what's special about Juanita is she is totally a lifelong learner, which is at the heart of what makes Trent University so special. And so after a career as an elementary teacher, she decided to come back and pursue further studies here at Trent. And when asked, why would you want to do that? She says, well, I wanted to continue to give a voice to those students who don't often have a voice of their own, or perhaps are afraid to use their voice. And I think, wow, there's very noble cause in that, and something so Trent and important for us to give voices and use our education to help those who can't have their own voice. And so without any further ado, I'd like to introduce Juanita Spears with The Power of Mirrors, Windows and Doors, Challenging Heteronormativity with the LGBTQ2S picture books in elementary classrooms.
I love a good story, don't you? Picture books, they're fun, interactive, and they really engage kids. They can be used as mirrors, windows, and doors. Mirror texts are books where children see themselves in the stories, and this reinforces that their narratives matter. Window texts are books that introduce cultures that are different from themselves, and this promotes understanding and an appreciation for the stories of others. Door texts are entrees into worlds that are truly different, perhaps even a bit scary or confusing, but this affords them the opportunity to overcome misunderstandings or biases. You know, we want our kids to be truly confident in themselves, tolerate differences, and have a strong sense of social justice. Building a sense of self, family, and community comes way more easily to children who see themselves in the stories that they read. Unfortunately, the worlds depicted in children's literature is overwhelmingly white, upper middle class, heterosexual, English-speaking, and male. When kids can't find themselves in the stories they read, or worse, they see images that are distorted, negative, or laughable, they learn a very powerful lesson about how they're devalued in our society. Unfortunately, research suggests that these picture books are not making their way onto school shelves, especially those that reflect the lesbian, gay, bi, trans, questioning, and two-spirited communities. I've spent the last 20 months doing extensive internet research, talking to Canadian publishers and authors, trying to find diverse picture books that have been published in the last 20 years and that reflect the LGBTQ2S communities and intersect with ethnicity and religion. I've only been able to find 25 books. I've evaluated them and assessed them, and I will be making their titles available on a searchable online database. You know, these picture books are vital in increasing the visibility of our queer youth, who comprise anywhere from 3 to 10% of our population. Can you just imagine if school libraries offered an equitable assortment of books that included all ethnicities, all genders, all diversities, how these powerful mirrors, windows, and doors could be used to develop an inclusive Canadian identity? A just society? You know, every child deserves to be seen, respected, and accepted for who they are. Their stories matter, and they should be found on school shelves. Thank you for listening to my story.
Thank you so much again, Juanita, for sharing your research with us. That was really inspirational. Thank you. Now, I would like to move on to another inspirational woman, a woman who really enjoys birds, really enjoys birds. And when she's not thinking about birds, she likes insects. And she likes them so much she likes to take photographs of insects. And when she's not taking photographs of insects, she likes to hang out with her pet tarantula named Bird. And so Jennifer Basie is a triple threat in environmental and life sciences, and she will be talking to us today about one of her interests, not bird, not insects, but in fact, wild turkey nesting behavior and genetics.
Hi everyone. Now, I study several different aspects of wild turkey behavior, but here I'm going to focus on some of their nesting behaviors and what those mean for the genetic composition of a wild turkey nest. Now, turkeys are non-monogamous, which means that during the breeding season, a male will mate with more than one female, and females will mate with more than one male. And this is a way for wild turkeys to sort of hedge their bets and ensure that even if, for example, a female has mated with a male that has really low quality sperm, she's mated with a sort of backup male and hasn't missed her opportunity to reproduce that year. Now, this means that in one turkey nest, we could see eggs that all share a mom, but have different dads. Now, turkeys also do something called conspecific brood parasitism, which is really just a fancy term for the fact that they will sometimes sneakily lay eggs in one another's nests. the conspecific part of the term referring to the fact that they are the same species doing this to one another, and the brood parasite part of the term referring to the fact that the incubating hen or the hen sitting on the brood needs to dedicate extra time and energy and resources into raising baby turkeys that are not her own. And so that's where the sort of parasite part of that interaction comes from. Now, while we don't know why wild turkeys do this specifically yet, we do know that in other bird species, they will sometimes do this when they perceive that there's a higher chance of their own nest being unsuccessful. And so perhaps they perceive that there are more predators in the landscape that year, or that nesting habitat quality is a little bit lower. And so studying these behaviors can actually tell us something about the environment as well. Now, in wild turkeys, this non-monogamy behavior paired with this conspecific brood parasitism behavior means that in one turkey nest, we could actually see eggs from different dads and different moms. And so we could have a sort of mishmash of genetics happening. My research over the past couple of years has involved finding these turkey nests in Peterborough County and collecting the hatched eggshells after the baby turkeys have hatched out and moved away from the nest. And then in the lab, extracting DNA from those eggshells and trying to figure out just how many males and how many females are contributing genetic material to each of those nests. Now, my samples are in the midst of being analyzed among this COVID-19 crisis, but I'm hoping that eventually I'll be able to figure out just how often Peterborough County's hens are sneaking around and laying eggs in one another's nests, and why they might be doing so. Thank you.
That was great, Jennifer. Thank you so much for talking about the nesting habit of birds. That was really enjoyable, so fascinating, so many great topics. And Jennifer was a triple threat, and I'm about to introduce another triple threat to you. Her name's Brittany McFarlane. Brittany is an academic advisor at Ontario Tech University down in Oshawa. She is in our educational studies program, and she's a mom. She has a wonderful 10-month baby who keeps her busy, she says, most of the time. And I bet she's keeping her busy right now. So without waiting too much longer, because I know you're on a schedule, let's go with Brittany McFarlane in educational studies with, I just have a quick question.
Imagine you are a university student who has been working through an issue for the past couple of days. Maybe it's personal, maybe it's academic. Either way, it's impacting your studies. Last night, you combed through the university's website to find more information, but you came up empty-handed and frustrated. So when you woke up this morning, you decided you just need a second opinion. So here you are. The name on the door is not familiar, but you're sure someone has mentioned this office to you before. Tentatively, you walk up, knock on the door and say, I've just got a quick question. Whether your question is quick or not, you have now taken the first step in finding support with an academic advisor. People hear the term academic advising and think that the main reason for this support is to help students register for classes. But in many cases, course registration is just a small piece of what academic advisors are doing every single day. So to develop a better understanding of what academic advising looks like across Ontario universities, I used a qualitative study to look at universities' public-facing websites to understand how are they describing academic advising. And then I interviewed 5 academic advisors to see who are our academic advisors and how do they do their job. And what I found is that advisors have a practice of advising. And this includes external and internal influences, which combine to create their philosophy of advising. They have strategies of advising, which is how they're interacting with students every single day. And then they use assessment to gauge how well they're advising students. And this advising practice is cyclical, and it's constantly changing based on the needs of students, the institution, as well as the advisor's own personal values, beliefs, and theoretical background. And understanding an advisor's practice is important because the quality of academic advising is dependent on who is in that role and how they understand their job. And so advisors have lasting and impactful relationships with students, but to have those relationships, advisors need to be able to reflect on their own advising practice and understand how their ideas and their potential can shape their practice of advising. As a member of the university community, you know that students have invested time, energy, and money to be here. And while students may share the same program with hundreds or even thousands of students, questions are going to arise as they navigate through this journey. So if you come across a student who is struggling, I hope that you will faintly remember academic advising as a support that is here to help, and you will encourage them, whether or not the question is quick, to knock on that door and say, "I've just got a quick question."
Wow, thanks so much, Brittany. That was so great to hear your presentation. Now we have another great presentation ahead of you from Elizabeth Pfeiffer. Now Elizabeth really loves comic books, and she's always loved comic books. And she comes to us after a career in journalism where she worked for many newspapers in and around Southern Ontario, and she's now at Trent University in our public text program. Now Elizabeth is married and has some kids, and so when I asked, "What do you do in your free time?" she's like, "Free time? What's that all about?" And I can understand what that's like, having some children myself, but it's even more impressive to think she's juggling family and studies and doing this all at this time of COVID, which is really lots of pressure on us all. So she really loves comic books, and she wants to share that love of comic books with us. So let's hear a little bit more what Elizabeth Pfeiffer has to say, part of our public text program with Non-Compliance and the Patriarchy, Bitch Planet, as a satirical exploration of gender constructs. Elizabeth.
The graphic narrative is extremely popular, and the multi-billion-dollar superhero industry that spans out of comic books is evidence to this. However, comic books have become more diverse and subversive as women, queer folks, writers, and artists of color are creating their own graphic stories. In December 2014, Kelly Sue DeConnick, also known as the writer who reinvented Captain Marvel as a woman, and Canadian artist Valentine Delandro created Bitch Planet. This feminist dystopia is what my research focuses on. It's so named because women are sent to an off-distant prison planet for any socio-cultural infraction, be they too black, too Latina, too queer, too trans, too femme, too butch, too masculine, too proud, too ambitious, or too political. As we know, the personal is political. It explores intersectional feminist issues through its main story, in which mostly non-white, non-compliant prisoners are forced to fight in a gladiator match. Think Hunger Games meets The Handmaid's Tale, with a bit of Pam Grier in the 1974 blaxploitation film Coffee, and a dash of Orange is the New Black. That top image you see there, right, that is from the Catholic nun, a holographic projection that greets the prisoners when they arrive at Bitch Planet. She's telling those prisoners, those wanted on offenses like wanton obesity, disrespect, patrilineal dishonor, murder, and trisomy 21, also known as Down syndrome. She's telling them on threat of death that their non-compliance is not recommended. Look at her, she's looking at us, the reader. That's right, because she's also telling us that our non-compliance is not recommended too. And that's because Bitch Planet is first and foremost a satire. It satirizes its own world and it satirizes ours through use of what we would call paratext. That's kind of like a fancy word for fringe around the text, like an inside cover jacket or a preface or foreword. In Bitch Planet, they use fake advertisements and essays on fat phobia, emotional labor, institutional and systemic racism, victim blaming, and sexual assault to explore intersectional feminist issues. The last image you see, right, those women running on a treadmill being told they're too fat because they're being blamed for their own body size and image. I think that seems a little bit familiar. The Peloton ad, anyone?
Well, folks, it's hard to believe we are now down to the last presentation of 3MT 2020. It has been an incredible event so far, so many passionate people, so many great topics, and now, last but not least, we have Sasha Noor. Sasha Noor is one of our environmental life sciences students. She is very upbeat, engaged in university and trail college life, and just an all-around wonderful person. She's from Ottawa, Ontario, by the way, and she has always been fascinated by the voices of small mammals. And when she's not listening to those little voices that come from small mammals, she's doing really fun things like gardening. She loves to garden. She loves to pursue a zero-waste lifestyle, which is pretty cool. And on top of that, she has, get this, an albino Pac-Man frog named Pineapple. I wish I could be that cool. I have a cat named Ginger, whom I love, but an albino Pac-Man frog called Pineapple, that just takes it up to the next level. So I think I hear Ginger's voice right now, so I hope that's not coming across on the recording, but let's hear a little bit more about other mammals' voices with Think Before They Squeak, vocalizations of the squirrel family. Sasha.
I want you to close your eyes, just for a moment, and think of the sounds of a rainforest. What are the things that you hear? Maybe the high-pitched songs of tropical birds, the loud yet familiar calls of howler monkeys, or the low roar of a waterfall nearby. Now, what about the things that you can't hear? What about the echolocations of thousands of bats in a nearby cave, or the low rumbles of a conversation between two elephants over 50 kilometers apart, or the chatters of a giant flying squirrel that patiently awaits sundown? Acoustic communication and vocal production in the animal kingdom is incredibly diverse, and we as humans only get to listen in to a small portion of it. Our hearing range goes from 20 hertz to 20 kilohertz. So my voice sits around 200 hertz, and it's related to how quickly the vibration of my vocal cords are. So the faster they vibrate, the higher my voice goes, and the slower they vibrate, the lower I can throw my voice. At the bottom of our frequency range, the sound waves get very large, and that means that we can feel the sounds better than we can hear them. These large sound waves are what allow elephants to communicate over a really long range, because that sound wave can cut through trees and other barriers in order to reach the receiver on the other end. At the higher end of our frequency range, we actually lose our ability to hear these high frequencies as we age, because our eardrum loses the ability to pick up these really tightly packed sound waves. So a baby can hear a tone of 20 kilohertz, but me as a 24-year-old can already only hear a max of around 14 kilohertz, mostly due to the loud music I listened to as a kid. Bats, on the other hand, can produce frequencies of up to 200 kilohertz, and they have specialized ears and nose structures that allow them to hear those really tightly packed sound waves and to use it like a sonar to catch insects mid-flight in total darkness. Now, what sounds have we missed out on, and how has the human bias impacted the way we interact with the rich acoustic community around us? That's where I come in. I study high-frequency vocalizations in animals, particularly in flying squirrels, and my thesis is dedicated into looking at how the way we interact with our soundscape impacts the way that we record animals and interact with them. So for squirrels, for example, we've recorded them for over 50 years, but we tend to use microphones that just encapsulate the human range. Therefore, we're missing the high frequencies, and it makes it really hard to figure out who's actually using high frequencies and why high-frequency calls are important in squirrels. So far, I found that social squirrels tend to use higher frequencies than their less social counterparts, and also nocturnal gliding squirrels use higher frequencies than those that are awake during the daytime, and I continue to investigate these in my ongoing work. Ultimately, my work calls for people to think about the types of ranges that they're using in their equipment so that they're encapsulating the entire range that an animal is using. In other words, I'm asking other researchers to think before the animals squeak. Thank you.
Wow, thank you so much, Sasha, for that presentation. And thank you at home for joining us for this special edition of 3MT, the COVID Home Edition. We really appreciate you joining us, and we really appreciate your attention. Now we have 8 participants, and now it's up to you to be the judge. Please see us at our website, www.trentu.ca/3MT, and make your vote known. You have until Sunday at midnight, that's the 24th of May at midnight, to place your votes. Then come back and join myself, the Dean of Graduate Studies, and the President of the Trent Graduate Student Association to give out the prizes on Monday, the 25th of May at 7 p.m. right back here on this website. Now, we have 2 prizes. In fact, we were going to give away 3 prizes. We thought that grad students really could appreciate this as the grand prize, but then we had second thoughts and thought perhaps that gives the wrong impression of what we're trying to do with 3MT, and by the way, it's far too valuable. So what we're doing is we have 2 People's Choice Awards. Please vote for the People's Choice Awards at the website that I told you about, www.trentu.ca/3MT. Thank you so much for joining us. Thanks to all the participants for coming out. It truly is a remarkable feat to do 3MT at any time, but there's extra pressure to do it at this time, where we're all separated. And remember, we may be separated, but we truly are together in our goals to pursue education, to make us better people. And I think there's no better way to show off what we do to the public than something like 3MT. We'll see you soon. We'll see you on Monday at 7 p.m. for the award ceremony. Thanks again.